So that brings us to today's lecture, and I'll just outline what we're going to go over, and what is kind of a must know, and what are other things to learn about. And so to begin this lecture, we're going to talk about different examples of RAS signaling in different signaling pathways. And so I'm going to go through the growth factor signaling, which is really a must know. This is sort of the paradigm for thinking about RAS GTPase signaling. But then I'm also going to give you other examples in which the RAS GTPase is used in signaling in different ways so that you can see how nature can build different signaling pathways out of these components. And the two examples I'll provide will be T cell signaling, which is very similar to growth factor signaling, but there are some differences there. And then I'll give you a very different example that can be found in yeast, where they also use RAS GTPases, but to signal in a completely different paradigm than what we've discussed so far. I'll then go over how our understanding of these different signaling pathways and the logic and control of RAS GTPases can help us understand how disease mutations work and why RAS signaling is so frequently mutated in things like cancer, developmental disorders. And then finally, I'll talk about some frontiers in RAS signaling. Uh, this is sort of, again, kind of an optional material, but if you find yourself very interested in RAS biology and you want to know more, you want to know where the field is going, we're going to discuss some interesting ways in which people have been approaching questions in RAS biology. We'll talk about some systems and synthetic biology approaches, including optogenetic control of RAS signaling, which is actually going to serve as a preview for the discussion paper we'll go through on Friday, as well as approaches to monitor effector use, so molecules interacting with RAS, systems wide using interesting mass spectrometry techniques known as BioID. And then finally, we'll discuss a little bit about how people have been trying to selectively target RAS, oncogenic forms of RAS, with drugs to try to ameliorate cancer. And there's been some new successes that target RAS biochemistry specifically. Okay, so the first RAS GTPase signaling pathway that we need to talk about is basically sort of the paradigm for RAS GTPase signaling that you really need to internalize for this class. And so, of all the things we talk about, this is the pathway you want to understand and know how it works. And what is growth factor signaling? Well, growth factors are molecules that are responsible for stimulating cell growth, proliferation, and differentiation of cells. And so essentially these factors bind to cells and through growth factor signaling will trigger growth, proliferation, and differentiation. And to just sort of see an example of this in action, if you take these PC12 cells and grow them in culture like this, untreated cells will adopt a sort of blobby morphology. But if you treat these cells with nerve growth factor, also known as NGF, it causes the PC12 cells to differentiate into neurites, like you see here. Um, so how does this signaling work? Well, basically, if you look in a book or at these slides, you'll see a diagram like this, and they say this is growth factor signaling. You've got growth factors, you've got a receptor, you've got GEFs, you've got a GTPase, you've got a kinase cascade, you've got phospho-ERK, and it's going into the nucleus. And to be honest, when you look at it like this, it can be a little overwhelming to understand what's going on. And so what we're going to do is we're going to step through the signaling pathway one step at a time so we understand what's going on at each step. And then after we've looked at it, we're going to kind of simplify uh, what's going on in the signaling into a couple modules that are connected together. And then it'll be really clear kind of how this pathway works. And it'll be easier to understand how other signaling pathways that use these components work as well. So let's start kind of breaking this signaling pathway down. So the first step in growth factor signaling is that growth factors will bind to and activate receptor tyrosine kinases. So you have these receptor tyrosine kinases here, and absent growth factor, they're inactive. But when your growth factor binds to them, it's going to trigger activation of the receptor, and these kinase domains become active. And these kinase domains will transphosphorylate one another on these tyrosine residues. And this creates what's called a phosphotyrosine platform, basically a whole bunch of phosphotyrosine residues on the cytoplasmic tails of these domains. And these phosphotyrosine residues can't really do anything on their own, But what they can do is create a platform for other proteins to bind in a regulated way. And in the case of growth factor signaling, there is a protein called GRAB2 that is able to recruit a guanine exchange factor called SOS to the membrane when these tyrosine residues on the receptor are phosphorylated. And the way that that works is that GRAB2 is a multi domain protein. Uh, and two of those domains, called SH3 domains, are responsible for binding to the guanine exchange factor. And another one of these domains, called an SH2 domain, is responsible for binding to phosphotyrosine on the receptor. And so this combination enables GRAB2 to serve as this adapter where the SH2 domains will localize GRAB2 to activated receptor. And when that happens, it brings this guanine exchange factor along for the ride. And so this is the primary way in which a growth factor is going to trigger activation of RAS. It's going to be through recruitment of the GEF to the plasma membrane. And that's because that's where the RAS GTPase is. So if you put the GEF and the RAS in close proximity to one another, you can drive activation of RAS. However, as we discussed in the last lecture, there are other domains associated with GEF proteins that are typically responsible for tuning their activity. And we actually talked specifically about the SOS-GEF in the last lecture uh, because there's a number of domains on this protein that are going to tune the activity of that core catalytic CDC25 domain. And so one of those is a signaling lipid called PIP2. And this lipid is related to how the cytoskeleton of the cell is being regulated. So different places in the cell membrane are going to have different quantities of this PIP2 lipid, and that's going to affect how much CDC25 activity is produced in the cell. And there is, of course, the domains here that interact with GRAB2. And so this is going to tune the activity of CDC25 in relationship to the magnitude of receptor activation. So the more receptor is activated, the more GRAB2 can recruit SOS to the membrane. And then finally, there's an interesting component of SOS, which is it contains a second binding domain that binds only to the activated form of GTP. And what this domain basically does is it creates positive feedback in the signaling system. So the more the SOS becomes, uh, the more SOS activity there is, that is, the more RAS GTP that's being produced by this GEF, the more active SOS becomes. And so there's a positive feedback loop here that enables this GEF to sort of amplify signals in the system. Okay, so once you've recruited SOS to the membrane and RAS gets activated, how does that continue? How does activated RAS continue to propagate signals? Well, as we discussed in the previous lecture, activated RAS can bind to effector molecules. And in this case, what RAS GTP is doing is it's going to bring an effector molecule called RAF kinase to the plasma membrane as well. And when it does so, it's going to trigger activation of that kinase. And the way that this activation is thought to work is that in the cytoplasm, RAF exists in a monomeric state in which the RAS binding domain and the kinase domain are sort of interacting with one another in such a way that the kinase has limited activity. But when RAF kinase is recruited to the plasma membrane and interacts with these RAS GTP molecules, that sort of pops the kinase open, freeing the active site of this kinase so that it can be active. And it also stimulates dimerization of these RAF kinase molecules, which helps hold them in an active conformation. Interestingly, the main mechanism of RAF activation appears to actually be the recruitment of the kinase to the membrane itself, because artificially recruiting RAF to the plasma membrane will activate it, even in the absence of RAS activation. And so it seems like when RAF is recruited to the membrane, it facilitates its dimerization and activation. And RAS is one mechanism by which you can recruit it to the membrane and do this. But if you artificially or synthetically recruit RAF to these locations, it will do essentially the same thing, drive activation of the kinase. So once you've activated RAF kinase, you're going to trigger a MAP kinase signaling cascade that's going to culminate in the activation of ERK kinase. So so RAF is what's called a MAP kinase. Um, and MAP kinases phosphorylate MAP kinases, which can phosphorylate MAP kinases. And so activated RAF will phosphorylate a MAP kinase called MEK, which will phosphorylate a MAP kinase called ERK. And if you're wondering why would you do this? One thing to recognize about this MAP kinase cascade module that you see here is that it can allow for signal amplification when you think about the stoichiometry of the different components. So there are about there are 10 to the fourth copies of RAF, the MAP kinase, but there are 10 to the fifth copies of MEK, and there are 10 to the sixth copies of ERK. And so a small number of molecules of RAF being activated can go on to activate many instances of the MEK, which can go on to activate even more copies of the ERK. And so whenever you see one of these MAP kinase cascade modules, you should just be thinking that the function is to convert a small amount of activated MAP kinase into a large amount of activated MAP kinase. In this case, the MAP kinase ERK. Once ERK has become phosphorylated, it will translocate into the nucleus. So normally that ERK is in the cytoplasm. Phospho-ERK will translocate from the cytoplasm into the nucleus, and once inside there, it hits a number of targets like Jun, Phos, ELK, and ETS, and phosphorylation of those targets is going to result in changes in transcription of genes that are involved in the growth, proliferation, differentiation, and survival of cells. And so, looking back at our chart, it's kind of like, oh, that was a lot of stuff that's going on. And it's a lot of words and it's a lot of names. And so it can be a little bit tricky to kind of internalize it or be like, how do I remember this? Like, how do I understand it? And so one thing that I think is really helpful when you see a signaling pathway like this or any sort of complicated signaling pathway is to kind of break it down into the core modules that are highlight how information is being transmitted in the signaling pathway. And so if we look at this growth factor signaling pathway, what we can see is it's really mainly built up out of three things. There is a receptor tyrosine kinase module. So this is the thing in which growth factors are going to bind and they're going to create these phosphotyrosine platforms. This module communicates its signals to the RAS module. So when this tyrosine kinase is activated, it's going to lead to the activation of the RAS module by recruiting that GEF to the membrane. And now, once this RAS module has become active, how does it communicate its signals? Well, it's going to communicate its information to this MAP kinase cascade module, which is what's going to ultimately lead to the changes in gene expression in the cell. And so, even though there's a lot of proteins and molecules here, um, and words and details, uh, at the same time, it's not that complicated. We've got a receptor, it gets activated. Activated receptor is going to drive activation of RAS, and activated RAS is going to drive MAP kinase signaling. So from this vantage point, it's pretty simple. And this way of viewing the signaling as being built up from modules can actually really help us understand why different outcomes end up happening sometimes when you use different uh, growth factor inputs to the cell. So, for example, there's this famous result that if you stimulate cells with NGF, nerve growth factor, and you look at ERK activation over time, you'll see this profile where ERK gets turned on and it stays on pretty much throughout the course of the experiment. And when this happens, it triggers differentiation of cells into one cell type. But if, in contrast, you stimulate cells with a different growth factor, EGF, interestingly, this produces a transient ERK activation. So ERK levels increase and then they decrease and they go very low again. Um, and when this happens, instead of differentiating, cells will proliferate. And so, why do we get these different outcomes, even though the overall kind of list of components between them is identical, except for the identity of the growth factor and the associated receptors. And so there is negative feedback, uh, interestingly, which downregulates receptor activity. So one of the things that distinguishes the EGF receptor from the NGF receptor is that phosphorylated ERK can go back and phosphorylate this receptor and downregulate its activity. And so this negative feedback means that as we produce more phospho-ERK, it's actually going to decrease the amount of activity that's driving its activation. And so if we think about it in terms of that modular kind of block diagram for the signaling, where we have the receptor tyrosine kinase module activating the RAS module, activating the MAP kinase module. In this case, we can create a new arrow which says the MAP kinase module is going to inhibit the activity of the receptor kinase module. And thus, even though there's a lot of molecular complexity, it's pretty easy to understand that this step is going to have the activity of downregulating the pathway. So, armed with this kind of like canonical now description of growth factor signaling, we are poised to look at some other RAS GTPase signaling systems that are similar to what we've discussed, but a little bit more different and a little bit more complicated. But hopefully, this idea of thinking about the pathway and breaking it down into pieces is going to make it easy for us to understand these more complicated systems. And so, what is T cell receptor signaling? Well, T cells are immune cells in your body, and their function is to recognize. One of their functions is to recognize virally infected cells. So, an infected cell contains a virus. The peptides from this virus will end up getting displayed on the cell surface. Uh, and T cells are crawling all out throughout your body, and they interrogate the peptides that are displayed on the cell surface. And if they detect something foreign, the objective then of the T cell is to kill the target. And so what you're seeing here is a video. I didn't record it, but what you're seeing here is a video of a T cell interrogating a target cell. And so uh, at the synapse between these two cells, there is T cell receptor signaling happening that's going to decide whether or not this cell should be killed or spared. And so T cell receptor signaling is involves RAS GTPase signaling just like before, but unlike the growth factor signaling, there are actually two distinct GEF proteins that participate together to activate RAS. And so one of these is the protein you learned about before, son of Sevenless, and the other is a GEF we haven't talked about yet called RAS-GRP. And these two GEFs together are going to be responsible for activating RAS in the cell. And so let's talk about how each of those GEFs are functioning separately, and then let's think about what happens when both of them are working together. And so the SOS branch of T cell signaling is very similar to the growth factor signaling pathways we discussed. That is, when the T cell receptor recognizes a foreign peptide, it's going to lead to the creation of a phosphotyrosine platform on a molecule in the cytoplasm. In this case, that molecule is called LAT. And exactly as before, the GRAB2 protein is going to interact with that phosphotyrosine platform, and it's going to recruit the SOS-GEF to the plasma membrane, where it can activate RAS and be regulated in all those different ways, like we talked about before, uh, like PIP2, feedback, and so on and so forth. And so, as I said, just like before, this is almost identical to what we saw with growth factor. It's just that instead of a receptor tyrosine kinase, it is a T cell receptor that's driving this assembly. Now, if we look at the RAS-GRP branch, we can see that it actually has a very different activation mechanism that enables RAS GTP activation to incorporate completely different inputs into the system. And so what's kind of interesting is that in this case, activation of the T cell receptor, as we mentioned before, creates a platform for phosphotyrosine. But in this case, that phosphotyrosine platform can recruit a protein called PLC gamma, phospholipase C gamma, to the plasma membrane. So instead of recruiting the GEF directly, we're recruiting this protein PLC gamma. And what PLC gamma does, and you may or may not have learned about this signaling molecule yet in the course, but it's going to convert the lipid PIP2 into inositol triphosphate and diacyl glyceride. So PLC is going to convert PIP2 into IP3 and DAG. And one of those, IP3, is a cytoplasmically distributed second messenger, and the DAG is a membrane-associated second messenger. And the interesting thing is that the C1 domain of the Ras-GURP-GEF interacts with the DAG, the DAG, in the membrane, and thus, as your DAG levels increase inside the cell, this allows Ras-GURP to be recruited to the plasma membrane, putting it in close proximity to Ras to activate it. And so Ras-GURP is being activated in a manner that's similar to SOS in that it's being recruited to the membrane, but the mechanism by which it's being recruited to the membrane is different. It's being recruited there not by phosphotyrosine directly, but rather by interaction with DAG. In addition, this soluble IP3 produced by PLC gamma will go on to trigger a release of calcium from the endoplasmic reticulum into the cytoplasm. And that elevated calcium is going to bind to domains on the Ras-GURP called EF domains that are going to allosterically increase its activity. Okay, so as these IP3 levels accumulate, they're going to trigger an efflux of calcium into the cytoplasm. That calcium is going to further activate the Ras-GURP protein. And so at this point, we have two different GEFs that are each participating and converging on Ras, but are regulated in different ways. So if we try to draw sort of that same block diagram as before, it will at least kind of help us understand kind of at a higher level what's going on here. So in T-cell receptor signaling, we have target recognition, which is going to trigger TCR phosphotyrosine modules. So target recognition is going to create a phosphotyrosine platform. With the case for SOS, this leads to the exact same logic we'd seen previously, that phosphotyrosine is going to activate a RAS signaling through the SOS-GEF, and that's going to go on and activate ERK signaling just like before. So this portion is almost identical to the growth factor signaling we saw before. But in addition, this phosphotyrosine module can communicate its signals to a PLC gamma module, which then produces the DAG and IP3, which goes on to activate the Ras-GURP branch of the Ras-GTPase signaling module, which also then will go on to stimulate activation of the MAP kinase module and ERK. And so this is kind of weird. Why would you do this? Why would you use two GEFs and that have different regulations and different properties to turn on the same target? Seems a little redundant. But there does appear to be an explanation, which is that if you look at the dynamics that happen from each branch separately during stimulation, and then look at activity in terms of how much phosphorylated ERK is happening in the cell. When you have both guanine exchange factor, both branches permitted to signal, what you find is that cells adopt this sort of bimodal ERK signaling response. So there's a subset of cells that very strongly turn on phospho-ERK, and then there's a subset of cells that don't very strongly turn on ERK. And so in this case, when you stimulate cells through their T-cell receptor, you get kind of this bimodal response. Cells are either on or off. And if you remove the SOS-GEFs from the system, and you stimulate, what happens is you get this sort of graded analog response. So you get a whole bunch of cells with different amounts of phospho-ERK, and the population doesn't divide strongly into cells that are strongly on or strongly off. Instead, you kind of have this distribution, this kind of graded distribution of outcomes. And if you remove Ras-GRP from the signaling pathway entirely, then you find that there's no response at all. And in this case, all the cells are off. So it's very clear that you need Ras-GURP to be able to trigger activation at all, but you need the SOS protein in order to produce this bimodal signaling response. And so the model for how this is working is that the Ras-GURP protein, if you look at its dynamics, it ends up being activated very quickly in this system. However, um, this then creates a pool. So RASGRP activates quickly. It makes a lot of RAS-GTP very quickly. And remember that RAS-GTP is a molecule that stimulates the activity of the SOS-gap. And so um, by generating this pool of RAS-GTP, SOS can be very potently activated, which is going to push a lot of molecules from being sort of this weakly on graded response to fully active. And why would you want to have this type of bimodal response? Why couldn't you do analog? Like, isn't it nice sometimes to be able to read out, different shades of gray? Well, if you think about the biology of what's happening here, remember that we're talking about a T cell interrogating a target cell. And the choice this cell is trying to make is, do I kill this target or not? And killing something or not killing something is a pretty binary process. Like, you can't half kill something or 25% kill something or 75% kill something. You got to make a decision about whether you're going to kill it or not kill it. And so the decision that the cell makes should be bimodal. The cell should say, I'm going to kill this thing or I'm not going to kill something. It should not be an analog thing. So it really has to make a commitment. And that's what the real consequence of having multi-GEF activation in this TCR system is. Okay, so there's one more signaling pathway I want to show you, just because the two examples we've talked about so far, growth factor signaling and TCR signaling, while different, are very similar in that uh, they both end up recruiting things to the membrane through a phosphotyrosine platform, and they both communicate to a phospho-ERK module downstream. But there are other ways in which RAS-GTPases can signal in totally different ways. And an example that we'll go through right now is yeast RAS signaling, which is in fact completely different from what we've talked about so far. And so I've got a diagram here of the RAS-GTPase signaling from yeast. And again, like so often it is with these diagrams, it's a bunch of words that we don't know exactly what they mean. But let's just highlight the key players here. So in this system, a protein called IRA12 functions as the GTPase activating protein for the RAS GTPase. And the CDC25, aptly named because you're familiar with that CDC25 name, this protein serves as the guanine exchange factor in the system to activate the GEF, or to activate the GTPase, excuse me. And in this case, what is the effector protein? So in the previous pathways, it was RAF kinase. In this system, the downstream effector is adenyl cyclase, which has the activity of converting ATP to cyclic AMP. And cyclic AMP, so basically in this system, RAS will activate adenylate cyclase to produce the cyclic AMP second messenger. And cyclic AMP can go on to activate protein kinase A and trigger a completely different type of downstream signaling process than what you get with phospho-ERK. And so, again, I don't expect you to memorize this, but it's just an example of how the same core GTPase, activation, inactivation paradigms and effector can be used to do something completely different from what you see in those TCR and growth factor signaling. So you might be wondering, well, yeah, but like yeast and humans, like, is it even really the same RAS? I mean, they're so different from us. There's been over 1 billion years of evolutionary divergence between yeast RAS and human RAS. Like, does it even make sense to talk about those RAS proteins as being similar to one another? And so, in fact, there's a really cool experiment that was done, which kind of confirms that these RAS proteins are not actually fundamentally different from one another. And so, what people did was they took the yeast RAS gene and they deleted it from those yeast, and they put back in the human RAS gene. So even though these proteins have been separated for over 1 billion years, they asked whether the human RAS gene could complement the activity of a deletion in yeast. And what they found was that the human version of RAS complements just fine. So in this assay, when you delete RAS, there's some assay in which the cells can't grow under a certain media condition. And when they have their regular RAS proteins in there, um, they can. And if they put in all these different human versions or different human isoforms of the RAS protein or viral RAS proteins as well, they are able to rescue these cells from death in a manner that's basically indistinguishable from the endogenous yeast proteins. And so wanted to highlight this so that it's just really clear to you guys that it's not the protein sequence of RAS, but rather the cellular context of RAS that's really important for controlling how it signals. So this exact same GTPase can be placed in this signaling pathway for growth factor signaling to connect growth factors to proliferation signals. Whereas in a T cell, placing that GTPase in this more complicated context can lead to an all or none binary killing, not killing type of signaling system. And that exact same protein placed into this totally different cellular context in yeast can produce something whose function is to relate nutrient conditions in the environment to growth conditions. And so I really want you guys to take away from this that it's not the protein sequence, but the signaling network and the context of RAS that's what's really important.